Hi, we're Group 8 of FIRE's HPI or Host Pathogen Interactions research stream. I'm Ray.
I'm Sophie.
I realized I was muted. I'm Chloe.
I'm Lexi.
For our research, we do exactly what our stream name suggests. We investigate how a virus or pathogen interacts with the host it has infected. Specifically, we're interested in the bacteria E. coli as the host and the T4 bacteriophage as its pathogen. A bacteriophage is just a virus of bacteria. Within E. coli, we're looking at a gene called thrA, which is involved in both directly and indirectly producing the amino acids homoserine, lysine, methionine, and threonine. Amino acids are used in bacterial growth.
So E. coli has around 4,000 genes that encode amino acids. Some genes are considered essential, meaning E. coli cannot grow without them, and some genes E. coli can find ways to cover the function that was lost. Our research, as Ray says, focuses on the thrA gene in E. coli. Determining essentiality of this gene is significant in research on the effects of different pathways in bacteria. Removing genes from E. coli not only hinders bacterial replication, but has shown to impact bacteriophage replication. Our project objective is to determine the effect of removing the thrA gene in E. coli on both bacterial and viral phage replication. This information can be applied to other areas of research on topics like drugs for bacterial infections, what amino acids are essential for both bacterial replication and bacteriophage replication.
So the figures below display the most important data from our research. They show the average T4 and T4R bacteriophage lysis rates, and this is the rate at which a virus is able to break down the E. coli cells. There are slower lysis rates seen when the thrA gene is absent compared to the parent strain. This could be due to the lack of amino acids present in the environment, because prior research has found that during viral replication, the level of amino acids increase. This means that when our gene is removed, it hinders the virus from obtaining the resources that it needs to effectively take over the E. coli cells.
So as seen by the data, the thrA gene replicated similarly to the parent strain in rich LB media, but slower in minimal media. And this means that the thrA gene is not essential for bacterial replication. There are important metabolites missing when the thrA gene is removed and the E. coli is unable to draw them from the minimal media. This also corresponds to previous findings that we saw in academic journals. And then when T4 bacteriophage is added, the parent and delta thrA plaque assays displayed similar formation, but when T4R was used, fewer plaques formed on the delta thrA assays. Finally, lysis rates are slower when the thrA gene is absent. So the next steps for this experiment would be to determine which nutrients the delta thrA strain requires to replicate while experimenting with why the missing gene affects bacteriophages and its ability to replicate and lyse the cells. And then finally, to explore whether it's possible to increase the growth rate of the knockout through supplying minimal media imposed with the amino acids that were found in the L-homoserine biosynthesis pathway.